Is there anyone in the room who doesn't know what this picture is? You all know what it is? What is it? What? It's not your thing. Okay. What? Of course, that's the Brooklyn Bridge. Now, you should read about the Brooklyn Bridge for two reasons. One, because it's probably the most famous bridge, at least in this country, if not the world. Linked Manhattan and Brooklyn in the late 19th century. And these fellows who were walking on the catwalk, ladies used to do that too, and have their pictures taken. Life was never the same after the bridge was built. And so that's the logo of the course. So visualize that we're in Manhattan and we're strictly basic reductionist scientists, and somebody else is over in Brooklyn and they're strictly clinicians and trying to come up with new therapies and for dread diseases. But they don't talk to one another if there ain't a bridge. And so one of the purposes of this course is to help to provide cross information to convey the excitement of the challenges. It's not the sort of a lecture where anyone expects that you're going to write down a whole bunch of numbers and facts and take a quiz on. And so we've been fortunate for the past 17 years now to have an extraordinary array of faculty, if you will, most of whom are here at the NIH, but others, like our guest today, Dr. Mellors, comes from another institution. Where possible, usually about half the time, we begin each session with a live patient. And the reason for it is, as someone put it I thought very wisely a few years ago, it puts a human face on a disease. And particularly for Ph.D. scientists, fellow students, and so forth. Turns out that's a pretty powerful thing, to realize there's a reality to what the words all mean. So, quiz. What global viral disease is apparently cured by engineered pharmaceuticals? Anybody know? It's a widespread disease throughout most of the world, millions and millions of people. A major cause of liver cancer, and it's not hepatitis B. What is it? Right, it's hepatitis C. So hepatitis C is, to my knowledge, the only viral disease causing serious problems in human beings, which is truly, it seems, cured by engineered pharmaceuticals, which interact specifically with different components of the virus. And that is a major, major advance in medicine. We've always had a session on hepatitis C. I don't think we have one this year, but you should certainly know about it. So that's hepatitis C, but today yes, sir. Oh, yes, yes. Well, that's a separate problem. So it's very effective, but the problem is that it came on the market at $80,000 for a 10-week course. Now it's down to $40,000. It's representative of the crisis that exists between science and the delivery of healthcare. That's a separate issue, which will come up to some extent today. So it's hepatitis C. It's not HIV, unfortunately. But HIV, despite the fact that it's been converted, amazingly enough, probably one of the great accomplishments in all of public health and medicine, it's been converted from an acute fatal disease, HIV/AIDS, into largely a chronic treatable disease. So as you'll learn, people live to be 50, maybe older years, as long as they take their medicines. But if that's the case, why are there millions of new cases each year? And why can't HIV, to the moment, be cured, like HCV, for example? And what happens if someone stops taking their medications? They relapse. Well, where is this virus? These are basic problems in virology and molecular virology and immunology. And so that's the domain that we're going to hear about today. And what does the future hold for regards to dealing with this disease? I would call your attention to, in just last week in the New England Journal of Medicine, there's an interesting editorial on the effect of one massive publicly supported program, the PEPFAR program, which Tony Fauci evaluated, and they pointed out that in I think it's 15 years now, or 5, I'm not sure, over 13 million people, mainly in sub-Saharan Africa, have been treated. It's estimated there are over 2 million perinatal HIV infections that have been prevented, and care has been provided for 6.5 million orphans and vulnerable children. So there's this linkage between the molecular, the cellular, the basic understanding of why, at the moment, we can't cure or prevent in an immunologic way, and the public health aspect of it on a global scene. So that's the bridge that we're going to talk about. And to talk about this, we are fortunate in having two truly global experts. John Coffin needs no introduction. He needs a medal. In the 17 years that we've had this course, plus 5 when I ran something like it at Tufts before coming here, John has been an active, almost annual participant. He never says the same thing because his research is really always at the cutting edge of where the basic problems are. So John received his Ph.D. with Nobel laureate Howard Temin at the University of Wisconsin. He trained further in Zurich and then came to Tufts in the Department of Molecular Biology and Microbiology, where he still is. But John has led a unique life because for the past, I think, almost 17 years, if not more, he has been commuting to Boston. I have a feeling when he shows up at the airport on a Tuesday morning, the airlines all bow down low because you must have the world's record. And he spends several days a week here and then also runs a highly productive lab at Tufts. He came here as director of the HIV drug resistance program at the NCI and continues in that area, advising, consulting, research going on in both ways. John is a global expert in retroviruses, their replication, and in recent years in particular, their evolution. He's received all kinds of honors as a member of the National Academy, et cetera, et cetera. And I won't belabor the point, but we're delighted that you're willing to do this once again. Next year, you're going to get a medal, I promise. Yes. Now, our second speaker is John Mellors. John is professor of medicine and has a chair at the University of Pittsburgh. The chair is for global elimination of HIV and AIDS. And so that tells you what his research and activities are involved in. He's chief of the Division of Infectious Diseases. He graduated from Dartmouth Medical School, trained in infectious disease at Yale, and has been collaborating and back and forth between Pittsburgh, Frederick, and so forth. And he and John have done some of the most important work that you will hear about dealing with these questions of why we can't eliminate this virus. So I think, John, you're going to speak first. So thank you very much.
Thanks, Wynn, and it's really a pleasure to be here. I learned today that my NIH badge was expired, so I had to go through the visitor center, which was interesting. So let's see. Where would my slides be? Here they are. Okay, great. All right, so Wynn raised the key questions. Why can't we eliminate HIV? And how does it persist, and what can we do about it? And this has, Demystifying Medicine will have two themes. So you get a 2-for-1 today. We want to examine how HIV persists both globally and individually. So first, global persistence of HIV. And instead of a case history, I want to preface some of the statistics that I'm going to show you by a quote that's inspired me as a physician throughout my career. Suffering, no matter how vast in number, is always individual. And think about that when I show you this slide, that there, as of 2016, almost 37 million people living with HIV infection. There's almost 2 million people infected annually with HIV and 1 million deaths, which is a significant improvement, but still a huge human toll. So for those of you who don't know, HIV can be transmitted most commonly by sexual contact, through pregnancy, childbirth, and breastfeeding, through injection drug use, and some poor public health planning and lousy leadership in our own United States can result in a nice, healthy epidemic related to injection drug use, particularly in the state of Indiana, where the thought of having a needle exchange program was felt to be immoral. You can get it through occupational exposure, and fortunately that's extremely rare, and blood or organ transplant, and that's as rare because of the extensive screening done of organ donors and blood donors. So what are the ABCs of stopping HIV transmission? Basically, the ABCs of stopping any epidemic is to block contact between the infected and uninfected individuals. But we're talking about a sexually transmitted infection. So stopping that type of contact is essentially impossible because our biology is programmed for us to reproduce and have sexual contact. So we need to reduce the infectivity of infected individuals. Okay, how do we do that? Antiretroviral therapy. Not only does it save the individual, but it saves many who come in contact with that individual. There were many cohort studies and then a randomized clinical trial known as HPTN 052 that showed greater than a 90% reduction in transmission from antiretroviral therapy that suppressed viremia. And possibly a cure could prevent HIV transmission and be part of the public health solution. The appeal of a cure is that one treatment would result in elimination of virus shedding and transmission. Eliminate contact with HIV. That's possible for blood supply. You can screen blood supply, and it's extremely rare that infected units are given to someone. And I believe in the United States that hasn't happened in many years. And as I said, preventing contact, sexual contact, is really not possible. Okay. But we can try to reduce the susceptibility of the uninfected individual. First of all, male circumcision reduces acquisition by about 50 to 60% in multiple randomized trials. It is being rolled out with varying enthusiasm by country and culture. Okay. chemoprophylaxis. In the HIV field, we call it this silly name, pre-exposure prophylaxis, or PrEP. But it's basically like malaria chemoprophylaxis. The uninfected individual who thinks they're going to be exposed goes on PrEP. And there's an FDA-approved 2-drug combination called Truvada or Tenofovir and M-tricytidine or FTC that I'll speak a little bit about in a minute. And then vaccine prophylaxis. We've had the hope and the promise of an effective vaccine ever since Bob Gallo isolated perpetually the HIV virus, and Margaret Heckler, the then Health and Human Service Secretary, said with great enthusiasm, we'll have a testable vaccine within a year. Okay, I think she was I think her statements were a bit misconstrued, and she's been pilloried for that one quote for many years. But it was an optimistic look towards the future. Okay, so what progress has there been in HIV prevention? Well, ART rollout through the program that Wynn mentioned, the President's Emergency Program for AIDS Relief, or PEPFAR, launched by George W. Bush, has been the singular global health success in the history of the United States. Okay? Billions of dollars, has resulted in declining incidence of HIV in several African countries, and I'll show you some of that data. Second, PrEP, if taken, works, and it works remarkably well. If you take it, the efficacy is thought to be in excess of 90%. And a recent study, the IPERGAY study, showed that on-demand PrEP, rather than daily, regardless of whether you're going to have a sexual encounter or not, on demand, 2 doses before, 2 doses after, had 90+% protective efficacy. That's led, after a lull following the FDA approval, of much more rapid uptake of PrEP, especially in the United States, and countries, progressive countries in Africa, like in Kenya, are rolling out PrEP as a national program. The good news is resistance so far from PrEP is infrequent. You see resistance mostly when you start the 2 drugs when somebody's already infected but not antibody positive, so-called during the window or eclipse phase of the infection, where the only way you can detect it is by quantification and detection of HIV RNA, which is not a practical screening test. And there have been some rare breakthrough infections in individuals on PrEP, and I'll tell you about one of those. But there's always a to and fro. With the uptake of PrEP, condomless sex has increased, and sexually transmitted infections are soaring, reaching an all-time historical high for chlamydia, gonorrhea, and syphilis last year in the United States. Okay, so that's the downside. Better PrEP is coming and is urgently needed. One of the kind of frightening things about sub-Saharan Africa is the youth bulge. There's an explosion of the population who are becoming adolescents, okay? And at this current rate of prevention and treatment, PEPFAR is not going to be able to keep up with that youth bulge. And so even though the incidence rates will fall, the number of individuals at risk will increase, and the total of number of cases could start to increase again in sub-Saharan Africa. So as basic scientists, if you want to work on something important, identify targets that are tractable to prevent HIV infection, and do it in a way that makes it easy, okay? By easy, I mean something topical, something that's long-acting oral, 1 dose for a month, 1 dose for a week at least, or injectable or implantable. And the holy grail would be an annual shot of a chemopreventive. Okay, that would be something spectacular. I could imagine a smart public health or Ministry of Health leader in South Africa saying, okay, every adolescent gets this annual injection before sexual debut through the age of 25. And that bar is enormously high because it has to be non-toxic, well tolerated, not painful to inject, does not interfere in any way with sexual reproduction or sexual health. And so there's plenty to be done in terms of discovering better molecules and better targets for HIV prevention. Okay, so here are the number of people receiving antiretroviral therapy, somewhere between 19 and 21 million. And this is since 2000. In 2000, there was a conference in Durban, South Africa. The president of South Africa, Mbeki, had colluded, and that's a very popular word. I think that will be in the final race to the word of the year with shithole. And I suspect the latter is going to win, but collusion, I think, will be a contender. So back then, Mbeki and Peter Duesberg teamed up and said that HIV was not the cause of AIDS. And this was a gigantic kind of coming out party and public lambasting of President Mbeki, and started a movement to treat the Southern Hemisphere, individuals with HIV infection with ART. And then George Bush jumped in, and now you have 20 million people. And it was said that this could not be done. It was also said when you could not cure hepatitis C, and it's being said all the time that you cannot cure HIV infection. So beware the negative prognosticator, excuse me. Okay, so these massive rollout has measurably reduced incidence and death from HIV infection. Okay, and this is only the front end, if you will. This is only treating the infected. Okay, here's the data from the IPER-GAY study. 2 tablets before, 2 tablets on the following 2 days after sexual exposure. Incidence rate in the placebo arm, 6.6 per 100 person years, reduced to 0.4. This is remarkable. Okay, and these individuals in this trial were having lots of sex. Okay, I mean, you get tired reading about the number of sexual encounters that they had. Okay, and here's the data on resistance. Rare in individuals who seroconvert while taking PrEP. 1 case possibly of tenofovir resistance, and a few cases of FTC resistance out of 160 seroconverters that were studied. Okay, and just an advertisement for our program that's funded by USAID called GEMS, which stands for Global Evaluation of Microbicide Sensitivity. We are the global center to monitor resistance arising from pre-exposure prophylaxis. Okay, what are the threats? That was the good news, but there's constant threat to HIV prevention. What are they? It's shown here on this slide, okay? This is the number of people living with HIV. This is who's aware. And you can see there's a gap of about 7.5 million. Of those who are aware, what proportion are on treatment? Another gap of 10 million, okay? But here's the most important threat that is a gap, and that's of those who are on ART, what proportion are suppressed? And suppression means that resistance will not develop, whereas non-suppression means that resistance will develop if there's drug exposure. And here is the threat of ARV resistance. And as a consequence of this, we see this slide. And the first rollout of ART was Tenofovir 3TC and a non-nucleoside RT inhibitor called efavirenz. And here's the prevalence of NNRTI resistance by region of the world over time since rollout, okay? And you can see this increasing incidence in Southern Africa, Western, Eastern, slower in Asia, Central and South America. And so we are losing first line ART, okay? Huge risk here. What's the solution? Well, we have TLD, which is Tenofovir, Lamivudine, and Dolutegravir. Better tolerated, higher efficacy than efavirenz-based regimens like TLE, Tenofovir, Lamivudine, efavirenz. Little to no transmitted Dolutegravir resistance so far because exposure is very limited. The beautiful thing is that ViiV and governments have negotiated to drive the cost of this pill to $75 a year, which is remarkable. I put $60 of gas in my Honda Pilot on the way down here, okay? All right. The thing that's a little concerning is it's being massively rolled out, first line, second line, and beyond. And so without much data, we're doing an experiment in somewhere between 10 to 15 million people, okay? What are the cautions? Well, it has been said by some, and some who'll rest in peace, that Dolutegravir was resistance proof. It is not. If you abuse the drug by giving it as a single drug, you will develop resistance in a significant fraction. The thing that makes me nervous is, although we have a new drug out there, we're recycling Tenofovir and Lamivudine, which was part of the first line therapy, and is PrEP. So we're using the same drugs for treatment and prevention. That's not a situation we want to be in. If we develop resistance from treatment, then you may develop breakthrough PrEP infection, okay? So it would be nice, again, to all you basic scientists in the audience, give us a little more in the way of options so that we don't have to use the same drugs for treatment and prevention, okay? And a double dosing of Dolutegravir is required when you treat tuberculosis with rifampin, and whether you know it or not, there's an evil kind of concordance between the HIV and TB epidemics. HIV is a formidable pathogen in terms of a bug that has caused more human suffering and death. Tuberculosis has to be in the top three and probably the top in the history of humanity. Okay, there are some breakthrough cases. They're rare. I'll just speed through this. This is a 43-year-old man who took his PrEP. That was proven by looking for tenofovir diphosphate concentrations in dried blood spots. He seroconverted. The virus he got had multiple NRTI and one NNRTI resistance mutation. These likely did not develop from the PrEP, but were transmitted. And so the conclusion was that the virus that he was exposed to broke through the PrEP, and so incident HIV is possible despite PrEP. We need to keep a careful eye on whether this continues. Okay? And again, raises anxiety about using the same drugs for treatment and prevention. Okay, but the good news is there's a relatively healthy pipeline of new PrEP. There's a dapivirine intravaginal ring that's under EMA review right now, which is female controlled and has that great advantage, empowers women to protect themselves. There's a different form of tenofovir, phenamide, that has less nephrotoxicity, kidney toxicity. It's in phase 3. Rilpivirine long-acting, which was recently, rilpivirine's approved for treatment, but the LA form was being developed for prevention, but it's been stopped because of cross-resistance and issues with cold chain. Cabotegravir, the daughter or son or nephew of dolutegravir, long-acting formulation is in phase 3. It's an injection, a large one, every 8 weeks, but gets us away from oral daily adherence. And there's excitement about 2 drugs, EFDA, that Merck has developed, and once weekly dosing can prevent macaques from SIV challenge. And there's thought of putting an implant with this very potent molecule in it that could last a long time. And similarly, Gilead has a remarkable capsid inhibitor program that's in preclinical, but the potency of this molecule is in the low picomolar, okay? And as a consequence of that, it's amenable to depot formulation. The mass required is such that you could fit it in a depot and put it under the skin. Okay, so what are the principles that we need to think about in terms of ending global HIV persistence? And this is a gross simplification. Each of these points is a study in and of itself. Recently wrote an editorial with Jean Nishega from South Africa, published in CID, about how we get that final group fully suppressed. So it's going to require, in my mind, massive continued rollout of simpler, safer, non-overlapping, and longer acting, both ART and PrEP, that will require new drugs and delivery systems, stronger public health infrastructure, national leadership and cooperation, which we have not seen a lot of lately, resources, monitoring capacity, and human persistence that exceeds HIV's persistence. So I'm going to stop there and see if there are any major questions before we move on to the second part.
Thank you very much, John. Please use the microphone. People who are watching also hear answers, but they don't know the questions.
Well, while everybody's thinking, I can ask a question.
Sure.
PrEP has any effect on pregnancy or sex health? In other words, in conception or anything association
Does
with?
not appear to. Birth control, on the other hand, seems to enhance modestly susceptibility to HIV. So we need really good PrEP to counter-effect the potential negative effects of contraception. The holy grail in the prevention field is combination prevention, multi-purpose prevention, a ring or an implant that could both prevent HIV infection and unwanted pregnancy.
John, would you comment about one of the effects of PEPFAR is the setting up of delivery systems of health care, of systems that didn't exist before in most of these countries. So isn't that a very positive thing in furthering these studies rather than expecting a big explosion with the bulge? It may very well be that this is a countering effect.
Health, strengthening health care infrastructure is essential. The question is it sustainable if PEPFAR, something happens to PEPFAR, and is it further expandable? Okay, and I can tell you at the level of Congress, there is no enthusiasm for expanding the PEPFAR budget. Okay. And it's undergoing some erosion, which means we're not well prepared for the bulge. If you speak to people in Botswana, in South Africa, in Kenya, who are managing these clinics, they are completely overwhelmed. The typical visit for somebody who's on ART lasts 7 minutes.
Any questions?
How much has it been eroded?
In real dollars, sure. The question is, how much has the PEPFAR budget been eroded? It's been flat. So there's inflation erosion. And we heard today, and Wall Street fell out of concern that interest rates are ticking up, and because inflation is picking up because personal wages are increasing. Okay, so the world economy has been good because money's been cheap. If money becomes less cheap, then programs that have a fixed budget and individuals who have a fixed budget will have less buying power. So. Yeah. Okay. Yep. So I'm going to continue to part 2. I'm just going to introduce part 2. Then John's going to tackle what he does best, which is cranberries and molecular virology. John's a cranberry farmer. And then I'm going to come back and pontificate or speculate or whatever you want to call it about how we might cure HIV infection. Okay. So a case of non-suppressible wild-type viremia. This was reported by Francesco Simonetti et al. And from Frank Maldirelli's lab, and really encompasses a huge group at Frederick who worked very closely together to get this done. But a 58-year-old African-American man was diagnosed with HIV in 2000. He was advanced at the time of presentation, which unfortunately is often the case, worse in minorities than in non-minorities. His CD4 count was 16. He had almost 300,000 copies per ml, but he was started on ART, and within 4 months, he was undetectable. His CD4 count rose, but never returned to normal, and that's one thing that we note, that the further advanced, and it's a common theme in any disease process, the later you treat it, the less well the body heals and responds to treatment, and this is no exception. Okay, after 12 years of reasonable health, his level of viremia started to tick up above the limit of detection of what's used to monitor in the clinic, which is somewhere around 50 copies to 200 copies. At the same time, he was diagnosed with squamous cell carcinoma of the tongue. Okay, this one was HPV negative. His therapy was switched based on a resistance test, but viremia persisted at about 100 copies per ml. And single genome sequencing analysis was performed in which individual viral templates were converted to cDNA, amplified, and sequenced. And here's the level of viremia that he experienced. He was undetectable. He started having onset of oral symptoms. His viremia peaked at between 300 and 400 copies, started to come down a little bit. His therapy was switched to include Dolutegravir, the integrase inhibitor. He suppressed about halfway back to where he was, but maintained a level of viremia that was detectable. And the single genome sequencing from the first time point showed a mixture of a virus with resistance mutations to his current regimen and wild type virus, and more wild type virus at the second time point without an intervention. And then after switching, the drug resistant virus disappeared, but the wild type virus persisted. And so this guy is going to tell us what's going on.
Thank you, John. And thank you, Wynn, for that overlong introduction. I guess the introduction probably doesn't need to expand every year that I give this, which has been, what you said, 17 years, something going approximately 17 years here, and then at least 5 years before that when we were colleagues at Tufts. So first I have to figure out how to get back to so I want to pick up from where John left off and talk about work principally actually that has been done in the DRP with a lot of important collaboration from John's lab as well as colleagues there up in Frederick, and getting at the question of why, in fact, we can't cure HIV infection. As Wynn pointed out, there's HCV, hepatitis C virus, is a very similar kind of infection to HIV in some respects. There's a development of enormous amount of genetic diversity, in fact, greater than in HIV. There is ongoing replication day after day after day, the same as an HIV infection. And with very recently developed drugs, the infection can be completely suppressed the way HIV infection can. But the difference is if you stop those drugs to an HCV infected individual, as little as 6 weeks after infection, having spent $80,000 on the drug, but that's another story, the HCV in 95% of the patients will never return. The patients have a very, very large fraction of the patients are in fact cured by this treatment. If you do the same thing with HIV, as John just told you, then in fact, even after 10 years or more of treatment with the virus load, which can be completely undetectable by standard virological assays. It's not always so, as in the case he just mentioned, but in most cases it is. The virus will inevitably return. There are very, very few instances of cases. There are a couple, but there are very, very few of cases where the virus suppression was permanent was at least long lasting after the interruption of therapy. Ordinarily, the virus comes back in a few weeks after you stop, and it comes back and restores rather quickly to very much the level of viremia, the level of infection that you saw before you started therapy. And so John showed you this picture before. The one thing I want to point out is that HIV, of course, is a retrovirus, which means it replicates through an integrated DNA intermediate in cells. And if you look, instead of looking at viral RNA in plasma, which is the standard way of assessing virus replication, you look at the virus DNA integrated into blood cells, peripheral blood mononuclear cells, or PBMCs, mostly CD4 positive memory T cells. You can see that the viral DNA persists at a much higher level than does the viral RNA. And within this DNA persistence, within this DNA, there are many, many defective genomes that occur during the course of reverse transcription, which is a little bit sloppy, but there are also a significant fraction of non-defective genomes that can give rise, if these cells are, for example, removed from the body and stimulated appropriately, to yield infectious virus. And so within those cells are the seeds for the return of the virus even after very, very long-term infection. So, so what's going on? Well, HIV presents us, the biology of HIV presents us with a few tools to study this process in rather nice ways, and I'll show you examples of how both of these are used. The first is that HIV is well known to generate a substantial amount of genetic diversity during ongoing replication. At the onset of of infection in almost all patients, in about 80% of the patients, as was shown originally by my colleague Mary Kearney up in Frederick, the virus is almost always a clonal infection. That is to say, the virus has been infection of an individual has been initiated by a single virus particle, at least genetically. So the virus is genetically uniform at the beginning of infection and then rather rapidly diversifies because of the error-prone nature of the replication process to yield a swarm of viruses, which if you isolate them after a year or two, you find no two identical sequences. And so you can use this process of diversification as a proxy for replication. The second useful property is that the viruses, retroviruses, which HIV is one, always integrate, must replicate by integrating their DNA copy of their genome called the provirus into the host cell DNA at one of a very large number of sites. So the probability of seeing two cells with integration at the same site is vanishingly small unless those two cells are descendants of one originally infected cell occurring by a process of cell division. So that's a useful process. And the fact that although you might think that integration is not necessary for HIV because it rapidly kills the cells it infects anyway, it turns out it is required, and the very good evidence for that is the fact that inhibitors of the integrase enzyme that carry out this reaction are very effective antiretrovirals. John mentioned a couple, Raltegravir, Dolutegravir, both inhibitors of this integration reaction. So these two, in recent years, there's been a fair amount of debate among between two possible models for persistence of virus all this time on therapy. The first is that the therapy is not quite as effective as we think it is, as it seems to be, that in fact there is indeed a little bit of ongoing low-level replication. It's that somewhere in the body, perhaps in some site like a lymph node or the brain has often been postulated for this, where ongoing replication can occur, but drugs don't penetrate very well. So the virus replication escapes the therapy in a site which is often referred to as a sanctuary site for replication. The second is that, in fact, there is no ongoing replication of any significance whatsoever, and that the infection is entirely carried forward by latent proviruses, which for some reason, which we don't really understand very well, despite some useful but probably not completely accurate in vitro models, process of latency in which they're not expressing virus, but those can occur in long-lived cells, and it's these long-lived memory cells. These are infecting immune cells, which have the property of turning into memory cells. These memory cells can then carry forward the infection for years and decades in the presence of a complete 100.0% suppression of the virus by antiretroviral therapy. So I'm going to talk first a little bit about the use of genetic diversity. So as I mentioned, infection is originally initiated in an individual by a single virus particle. Initially, and if you look very early in infection, you see the population is genetically quite uniform, but with time it diversifies, and after a while you get this very nice diverse population. And so you can see this nice genetic tree, and I'll show you a lot of these trees as we move forward here. And what you can see in this is that there has been a lot of diversification of the virus, as indicated by the length of these branches in this phylogenetic tree. The alternative is that there has been, instead, that if you introduce antiretroviral therapy, then you stop ongoing replication. You get and so you can get in proliferation of individual cells which have a single provirus, and therefore the production of a genetically identical population of virus And that leads to what we refer to as the clonal expansion of a single viral genome sequence that you could then detect by, again, the sequencing analysis of viral RNA in plasma or viral DNA in infected cells. And so this shows one patient, for example, at where you can see before therapy in the hollow symbols here, there's a very diverse virus population. And this is now plasma RNA. But if you look at even fairly short times after infection, you start to see the appearance of these clonal populations, both the viral DNA in black here and of viral RNA, despite complete suppression down to undetectable levels of RNA here, you can still isolate some RNA from plasma, sequence that, and see the appearance of this expanded clone of a single infection. But you can also see that over this time on therapy, there has been no increase in the genetic distance of any of these sequences from the starting virus. Importantly, if you take this is another 2 patients, and actually stop therapy, in this case at 7 years, in this case at 5 years after initiation of therapy, again you see no increase. Again, the pre-therapy samples are in hollow symbols, and the rebound virus after therapy is stopped are in the colored symbols. And you see, again, no increase in the genetic distance, in the overall genetic distance, again suggesting no replication of virus. But you do see the expansion of specific sequences into these clones, again implying that there has been a process which has allowed allowed clones of virus to expand. And this observation alone could be explained in 2 ways. One is the idea we favor, and I'll present evidence for in a minute, that in fact what's happened here is that single infected cells that are making virus have themselves clonally expanded by cell division, and were in fact infected before therapy was initiated, were during all the period of therapy were not just sitting there, but in fact were dividing a bit, so that, and either continuing to release virus on therapy, as we saw in the previous slide, or at least were the virus that they were making was allowed to replicate once therapy was resumed, was interrupted in the patient after 5 or 7 years here. But the alternative is that there was some small ongoing clonal replication in some sites, which leaked through and provided the virus that then rebounded, a lot of the virus that then rebounded later on when you stop therapy. To distinguish between those, though, we could look then at 2 things. Well, first, to ask in a more incisive way whether there was any evidence for ongoing replication. The previous study started with patients from which the virus was initially quite diverse. And that provides a lot of background, and it's much harder to detect additional diversification with any sensitivity whatsoever. But if we were to start with individuals who have been infected very, very recently, look at the virus there, that is much easier to detect even a small amount of additional accumulation of mutations and diversification. And this was made possible by a collaboration with Gert van Ziel and colleagues at the Stellenbosch University in South Africa, through Mary Kearney up in Frederick, and actually mediated by John Mellors when this started out, was using children from South Africa who initiated antiretroviral therapy in very early infection. Now, you can't do this experiment in the United States anymore, thankfully, because there's virtually no mother-to-infant transmission in the U.S. It's very, very rare, but there's still a lot in African countries. And so we know pretty closely the time of infection of these children, which is usually around the time of birth, or rather shortly before that. And at that time, the virus that you get tends again to be initiated. The infection tends again to be initiated by a single particle. And therefore, you start with much more uniform population. It's much easier to see diversification occurring on top of that. So here's a case first of a child who was was treated, but was treated fairly long after the initial sample was taken. So one sample was taken very early in life, and then there was a year or so from the time that sample was taken until therapy was initiated. And so you can see the orange symbols here indicate plasma virus that was taken before antiretroviral therapy, and the black triangles then after 6.9 years on antiretroviral therapy, but the first year of that was without therapy. And you can see that most of the virus, in fact, has diversified in a significant way in this child. However, if you look at a bunch of other children in this study, and I'll only show you one, this is one where our antiretroviral therapy was initiated quite early in infection and quite soon after the initial sample was taken. And what you can see here then is that here's the virus early in infection. It was almost completely uniform with a little bit of diversification, but really very, very little. Almost all the virus was in this completely uniform rake of sequences, identical sequences. And you can see that if you look now 8 years later, the DNA in this virus most of it was still absolutely the identical sequence, indicating that there had been no diversification, and the little bit of diversification there was not different from what was seen with the RNA early on, implying that there had been no additional detectable initial replication that would have allowed for the diversification. I will admit that this is not a tremendously sensitive assay. You could have a number of cycles of replication before you start to see diversification, but it certainly implies that ongoing replication is not an important factor in the maintenance of these viral sequences, that these viral sequences were maintained largely by persistence within long-lived or replicating cells. And so with this kind of evidence, there's many more studies have been done that give exactly the same result. There's no evidence for ongoing cycles of replication, and that implies the reservoir is not maintained this way. Although others have proposed now that it's not seen in blood, but it can occur in the lymphoid compartment, for example, in lymph nodes. So what's going on there? So Mary again set up a collaboration with Steve Deeks' group in the DARE Collaboratory headquartered in UCSF, where she obtained lymph nodes and blood cell samples from a number of suppressed individuals and compared them to pre-therapy populations in the same way you just saw with blood cells, and also took some longitudinal sequences. So here's one example. The hollow symbols in this and the subsequent slides will be the samples before antiretroviral therapy. Again, you can see this diverse population. And again, you can see that in PBMCs, as before, there's no subsequent diversification of the virus, and there is the expansion of actually a detectable, even pre-existing clone in the blood cells. But you can also see in the lymph nodes exactly the same sequence. There is no distinction between the sequences you see in lymph node, even including this clone, and what was present in the blood either before or after 5 years on suppressive therapy. So it's in general the same result as we saw with the PBMCs alone. And furthermore, if you take samples at 2 different times from lymph nodes, it's about a year apart after 4 and 5 years on therapy, and you just look at these 2 shades of blue, you can see again these samples are completely mixed. You'll notice, by the way, when you look at DNA, you see these hypermutant sequences. These are caused by the action of an antiviral factor called APOBEC3, 3G or 3F, that causes mutation of the DNA to a considerable extent. You'll virtually always see these in collections of viral DNA samples. And for the purpose of this talk, we can just ignore them here. They lead to very highly defective viral DNA. So, and finally, if you look at two different lymph nodes in the same individual at the same time, left side and the right side, again, the two different shades of blue here, you can see that these populations are really completely mixed. There's no evidence whatsoever that there's any compartmentalization, at least in this tissue. And Frank Maldarelli and the group has looked in autopsy samples at DNA sequences from viral populations all over the body of a few infected individuals and found the same general result. There's no evidence for any compartmentalized replication. The one exception might be occasionally in the brain, but those viruses don't seem to get back out into the general body when you can see the different independent evolution there. So there's no evidence for ongoing cycles of replication in lymph nodes of adults fully suppressed on antiretroviral therapy. No evidence for compartmentalization among lymph nodes in peripheral blood, despite some claims you'll see actually in the fairly recent literature if you look. And so we conclude from this that the HIV reservoir is not likely to be maintained by ongoing cycles of virus replication. And the issue is important, and John will come back to this when he talks about how you might cure HIV infection. You have to deal, so to cure HIV infection, it appears from this that simply developing more potent antiretroviral therapy will not be curative in this respect, because it is clear that at least the major, perhaps there's some minor component of replication, we can't say there isn't for sure, but clearly the major component is the persistence of viral DNA in long-lived, perhaps replicating cells. And without dealing with that, there's no hope whatsoever for curing HIV infection. So it's perhaps, I would say, slightly possible that if one figures out how to deal with that, there might be an issue of ongoing replication, but I think it's very, very unlikely. And so the focus on curative therapies has to be in these long-lived cells or stopping the virus that's coming out of them, one or the other. So it was very clear that you could see the appearance of these identical clones of sequences, or rakes of sequences, we call them. So do they come from, they are perhaps identical, some of them are maybe left over from the very early stages of infection, or they may in fact be, as I said, expanded cells from clonal expansion, from clonal replication of cells. So to look at this, we can go to we will go in a minute to looking at the role of integration sites, but first just let me kind of recap where I think we are at this point. So there is some kind of a persistent steady state. Remember, the amount of DNA remains quite constant in infected individuals after an initial decline on therapy. And the amount of very low level RNA, when you can measure it, also tends to remain highly constant, maybe with a slight decay with time. But so what do we think is happening? So in ongoing untreated HIV replication, you get most of the replication that's going on is in short-lived cells that are infected and die within a day or two of infection, and a small population of long-lived cells. So it's these really, though, that carry forward the infection day after day, and these are actually, I think, a virological accident in a sense. I don't think they play a major role in the natural biology of HIV infection. But what they do is foil our attempts to cure it. So after treatment with antiretroviral therapy, you get rid of all of these short-lived cells. You're left with the long-lived cells. And then with time, what we see is the appearance of these expanded clones, but the amount of DNA remains the same. And so from that, there have to be two processes going on. One of them is erosion, the loss of cells containing some proviruses with time. And the other is proliferation, the expansion of certain clones. And so the net result of these two processes, the overall amount of DNA remains quite constant in infected cells. If that's the case, we ought to be able to detect this proliferation of cells by looking at integration sites. And let's just talk about integration sites for a minute. So HIV irreversibly integrates at one of many millions of sites in the cell genome. The number of integration sites possible is really very, very large. Sites of integration then can uniquely tag individual cells and their progeny. If you can detect a provirus and its integration site together, you can, and you see that same identical integration site in two cells, those two cells have to have come from been the same cell at the time that the infection occurred. Okay, it's a very important conclusion to be able to make. In long-lived cells, sites of integration are determined both by the initial preferences as well as possible selective effects that that provirus might have had on the cell's ability to grow and survive all this time. And also, also by considerable effect of just chance of just being survival, the luckiest in a sense. So the assay we have used to assess this was originally developed by Rick Bushman and Charles Bangham, involves sharing infected DNA, ligating linkers, PCR amplification, and paired end Illumina sequencing. So from one end of the Illumina sequencing, you get the integration site. You cross from viral into cellular sequence, and it's always the same. Retroviruses always integrate the DNA exactly the same way. So the ends of the virus are always the same. So you can use that fact to be able to exactly identify where in the cell DNA the virus went in. And then from the other end, you can tell where the DNA was broken when you sheared it. Now, since you can only get one end on each DNA molecule, if you see two different shear points on the DNA, two different break points on two different DNA sequences after you PCR amplified this whole gamish, then that must be two different DNA molecules that were present in your original assay mixture, and that must have come from two different cells. There's no way those two sequences could have come from the same cell. So, so multiple sequences with the identical integration site and multiple breakpoints imply clonal expansion of the cell after infection. So we've assessed in a number of experiments, we've assessed the integration site distribution in several different cultured cells, including freshly isolated blood cells, CD34 positive hematopoietic stem cells, and a variety of cell lines, running about 150,000 to 300,000 sites in each sample. So that's 10 sites per gene or so. So it's a very nice, rich sequence from HIV infected and also from HIV infected patients during suppressive therapy, and compared all of these using an RNA-seq assay with the gene expression over the whole genome in each of these cell types. So in the next few slides, I'll show you cumulative integration sites in over small intervals of a particular chromosome, and you'll see at the end why I chose this particular one, chromosome 16, sort of a middling-sized chromosome. And the integration sites will be shown as red bars, and the expression level of each gene will be shown in a pale blue color. And as you will see, the distribution of integration sites is highly similar among all these cases, even between freshly isolated blood cells and highly aneuploid epithelial cell lines like HeLa. So even in very different cell lines, the distribution of integration sites really is remarkably similar. And this just shows you, so this is all of chromosome 16. That you can see the dirty little secret here, by the way, of the complete total genome sequence. And that is in every genome sequence, there's a huge chunk of DNA around the centromere, which is completely unsequenced because it's very, very difficult to approach. There's all kinds of repeats and things in there. So this is just a sort of even a guess as to exactly how big the sequence is. But what you can see if you just look down the ex vivo ones, the ones that were done in cell culture, including the PBMCs, which are down here. These are 293 cells. They're a kidney epithelial cell line. HeLa cells, which, as you know, comes from a cervical carcinoma cell line, very highly aneuploid. It's been in culture for many, many years. And then Jurkat cells, which are a cell from a CD4 positive T cell tumor. So these are malignant cells, a cell line, and as are these. And these are the PBMCs. And what you can see, actually, just looking both at the gene expression, which is the shaded blue areas, and this is over the whole chromosome. So each little line here, each pixel is basically 350 kilobases, and this will get more and more refined in the next few slides. But the overall impression is very strongly, very high level of similarity, identical clusters of integration sites, and that holds true almost no matter where we look. We can see, if we really bore down into this, we can see an occasional gene where there's a big difference in integration frequency, and almost all the time that correlates with a big difference in expression level of that particular gene between these different cell lines. But actually, sites like that are quite rare, and almost everything, if you look again, you can see. So what I've done here then is expanded the boxed region in the previous slide and tenfold, and now you can see again exactly the same thing. This impression, we're getting down to where we see some individual genes at least coming out from this. And finally, if we bore down one more level, we do get to the individual gene level. And again, you can see the very strong similarity, very surprising to us actually when we first looked at this, because these are very different cells in genes. And here's a gene called MKL2. And the reason I show you this sequence now here comes from, not from cell culture, but from cells directly isolated from patient 1 that John introduced to you. And what you can see here, you can't quite read the numbers, but here's the number of integration sites in this one very small bin right here. This bin is only 4 kilobases, and you can see almost all the integration sites are clustered right in this one region. There are only 1,000 integration sites are so isolated from this patient in this experiment. About 3 or 4% of them were right in this one small region. And here's that one gene, MKL2, which is a semi-oncogene. So it has been seen, some rearrangements in this have been seen in some tumor-like conditions. And what you can see is that if you look in the PBMCs, you can see integrations all over this gene, although it's not if you look, can I get back one? It's not a high frequency gene. This other gene even right next to it here is actually a better target than is this gene. And this, the region that's seen amplified here, a frequent target is not the high target area of this gene. So if you look in the in PBMCs, you see the integrations are in both directions, as indicated by the different color and different pointing arrows. And but if you look in our patient 1, what you see is a cluster of integration sites, independent integration sites. There's 16 different integration sites here, all of which are pointing in the same direction. This actually, if you're an old-time retrovirologist like me, this is a very, very familiar type of picture. This is what's seen in retroviral-induced cancer. It's the reason that retroviruses were actually first of interest for scientists. The reason why HIV was actually discovered within the National Cancer Institute and was, or discovered or not, as you wanted to interpret it, but it was originally first studied in the National Cancer Institute in the US, was because retroviruses were studied as very important models for cancer. And one of the major ways they cause cancer is by integrating it, by by selection of chance integration sites in the vicinity of the start sites or within genes called proto-oncogenes whose misexpression can send a cell down a path to becoming a cancer cell. And this looks very much like that, although this is by no means a cancer cell. These cells were only a very small fraction of the total cells. There's no evidence that they were expanding into a cancer. But there has to have been, the only explanation for seeing this pattern, this is not a hotspot for integration here necessarily, but it is a hotspot for selection of this cell after integration in the context of an HIV-infected and antiretroviral therapy treated patient, was to give rise to some change in the expression, the protein structure. We don't know yet. We're working on that, of this gene, and that must have caused the cell to replicate a little better than its neighbors or to survive better than its neighbors, and persist for a long time. We've seen several other genes in which this is the case. But by and large, you see that the sites of integration seem not to be in interesting genes. They seem to have merely been chance events that in some cases were subsequently amplified, probably by the cell responding to homeostatic signals or responding to specific antigens to which they happen to be reactive. So, but in this case, integrations in this MKL2 gene, along with a couple of others that we found, they're clearly due to selection or for preferential growth or survival due to the effects of the provirus on gene expression. So if we look at the overall distribution of integration sites, just by counting numbers of breakpoints, in this sample, small sample that we took from this patient, about a million or a few million cells, we can see about half the ones we saw only once. About a quarter of them were seen expanded, but fewer than seven times, so we don't separate them out. And then another quarter in aggregate were expanded more than seven times. And some of the ones were some of the integrations in the MKL2 gene here, a couple more down here. But the most prominent ones were not in MKL2 or in any other oncogene that we know. One of them in a site that we actually is repeated several times in the genome, so we couldn't identify specifically where it was. Another one in an interchromosomal site, we know where it is, but it's not near any gene. And then a bunch of others. But let's talk about this. It's one in this ambiguous site, which we called AMBI1 for that reason. And but before I do that, let me just address one other question. So we see a lot of this. A lot of the integration sites that we see, as I said, have been in cells that are clonally expanded. And the question of interest is all of them in clones. Is it only possible to see ones that have, in fact, been able to clonally expand all this time? Well, we think that's the case, actually. We believe that all of the ones that we see have been clonally expanded to some extent. But our sampling is only a few million out of something close to 1 trillion CD4 cells in the body. So our ability to actually infer that from the sample we can see is very limited. And of course, we don't know. We could make guesses as to what it is, but we don't know what the underlying distribution of clones it is. It's a very difficult problem. For example, if any of you have been to Boston or lived in Boston, you might recognize this particular scene. This is the Prudential building, which famously has a light on the top that tells the weather, although you wouldn't have been able to tell that from this scene because it was doused in fog one morning last fall when I took this picture. But so what does the rest of the city look like? That's what the problem of trying to infer what the clonal structure is from the few small sample that we can see. Well, there's the rest of the city. We can tell that from looking when the fog's not there, but we don't have the ability to do that when there's snow. When we can't sample it in any useful way. So this clonal expansion of affected cells, you can imagine, can arise in several ways. One is direct effect of the integrated provirus on cell multiplication or survival I just talked about. also chance outgrowth of memory cell during homeostatic replacement, and also antigen-driven expansion during the active immune response. And probably both of these things are taking place, at least in some patients. So, but those have nothing to do with the provirus, but it serves to mark all the descendants of an originally infected cell. So they can serve as markers, but they're not probably affecting the biology of the cell in any important way. And that's probably true of the vast majority of them. But there has been a popular hypothesis that much of the reservoir in infected cells, the reservoir that gives rise to the virus after therapy is stopped, is in resting CD4 positive cells. By resting, we mean they're not dividing. And the idea had been that activation of such cells to divide by antigen, homeostatic, or other signals will also activate expression and production of cells with non-defective infectious proviruses. And you can do this in vitro. You can pull these cells out. This is an experiment that John Mellors does with great frequency. You can pull these cells out. You can treat them with various agents that activate these cells in this way, and and get expression of infectious virus that you can then study. But the hypothesis has been that such cells will not survive this process. They'll die due to virus replication, and therefore, or the immune response to them, and therefore, you can't have clonal expansion of cells that contain infectious proviruses. And so the next few slides, and the last few slides I'll show, will be one actually falsify this hypothesis. This actually is patient one again, just shows the course of treatment of this patient and the level of viremia after John already presented him to you. In words, this is just a picture that shows what he said before. That is to say, the patient started with a very high level of viremia and a very, very low level of CD4 cells, and then went on with a partial restoration of CD4 cell activity and a complete suppression of the virus, except that he lived in an area where sometimes getting drugs was kind of difficult, and so he had a couple of interruptions in therapy along the way, but even after both of them, he was restored to full suppression. And then toward the end, and this is the slides that John showed you basically before, there was still, while still on this previously suppressive therapy, the virus load crept up. The therapy was then switched. It came back down part way, and you could see, as he said before, the disappearance of this diverse drug-resistant virus resistant to the first therapy, not the second, and then the survival only of this wild-type virus population, suggesting that this population of genetically identical virus may have been the product of a clone of expanded cells. Well, we got lucky in this case. We were actually able to isolate the AMB1 provirus. We knew its integration site, so it was a fairly straightforward PCR experiment to use a flanking primer from host DNA and some primers from internal primers from the virus sequence and amplify the whole provirus in two overlapping chunks. And what, in fact, John did in his lab was then to take these two chunks, mix them together, transfect infected cells. When you do that, you get some recombination between these fragments. Howard Temin showed that many, many years ago, actually, that you could do that, and was able to rescue the virus that came from this. So this virus was, in fact, fully intact by sequencing, and was shown in his lab to encode fully infectious virus. So that actually falsifies the hypothesis that I expressed before. And as it turns out, if you look at the sequence of the virus in plasma and the sequence of virus that's, so if you look at the sequence of the virus that's rescued from this transfection, or just sequence that you got from the original PCR, and you look at the sequence of this predominant virus in plasma, they're exactly the same. So this clone of cells is producing the virus that comes out in plasma at, in this case, about 100 copies per milliliter. A persistent production of virus from this one clone of our cells with this one integration site. And so far, this is the only clearly known case. Now, there's some labs, including John's, have also identified a number of other individuals where you could take individual cells and rescue from patients on therapy for a long time, rescue virus from those cells individually, find that a lot of the cells are producing, again, exactly the same virus. That virus that they're producing is exactly the same as the predominant virus you can see in the blood of those patients at the same time, strongly suggesting that this is a rather general phenomenon. But in those cases, we still have been, and we have still been unable to identify the specific integration sites firmly. That's still in process. But almost certainly where you see identical sequences in such an experiment in patients who've been infected for a long time, that's almost certainly due to clones of infected cells, the same as this. But absolute proof of that requires obtaining the integration sites, and that will happen. It's just taking a while to do so. One of the points of this is that if you now look, this is an assay that Mary Kearney developed where you can look at both DNA and RNA in cells and ask what fraction of cells that contain a specific provirus are also potentially producing virus by being able at the same time to express virus RNA. And I'm just going to refer you, these now are sequences, viral DNA sequences from this patient, and then aligned with independently obtained viral RNA sequences from individual cells of this patient. And in this particular case, the AMB1 proviruses, they were 384 AMB-infected cells were assumed to have gone into this assay originally from a count of the number of DNA molecules in such an experiment. Only 4 of those had detectable RNA sequences, and none of those at very high levels. So only at any one time, only a very small fraction of these cells in this case, so there's some kind of latent infection in these cells that most of the time does not allow their RNA expression. Only very rarely can such cells, I'll get to you in just a second, only very rarely can do such cells make virus. But yet there's some cells somewhere with this clone that are making enough virus to be detectable in blood, which is actually a fair amount if you think about it. Even 100 copies per milliliter of blood multiplied over the whole body, and then considering what's in the tissues as well, it's quite a bit of sequences. So somewhere there's probably a very small fraction of these cells that are making very large amounts of RNA, enough RNA to be making significant quantities of virus, but we haven't found them yet, despite having looked at literally thousands of sequences from these in this kind of an experiment. And so the model here is that cells, what happens is you get these clonal expansion of cells where the virus has gone into some kind of latency that we don't understand. Understanding this mechanism, this latency, is a major goal now of HIV cure research, because only by understanding it can we come up with ways to reverse it effectively, as John will tell you in just a minute. And then occasionally one of these cells, for some reason, will start to produce some virus RNA, and that then, of course, you're still on therapy here, so the antiretroviral therapy will block the appearance of new infections. But this virus is always around. So as soon as you stop therapy, this will come back and take over the population. And that's why you see this clonal outgrowth of viral sequences after you stop therapy. Unfortunately, we're not going to be able to do that in this patient because he died a couple of years ago from the squamous cell carcinoma that John mentioned. So we find no evidence for either so to summarize, this concludes what I have to say. We find no evidence for either ongoing replication or compartmentalization of HIV in blood or lymph nodes, implying that infection is primarily maintained as long-lived cells infected prior to therapy. Integration sites can be readily identified, and The integration site may, although I think this is a fairly rare outcome, but it is clearly a detectable one, may influence the persistence in clonal expansion and therefore the virus that rebounds. Clonal variants in plasma can be mapped to individual integration sites, and expanded clones can contain infectious proviruses. So this study was done in collaboration with very importantly in collaboration with John Mellors' lab, along with the collaborators at the HIV drug resistance program, particularly the labs of Mary Kearney and Frank Maldarelli, and the lab of Xiaolin Wu, who did all of the integration site analysis work. I will close with that and return you to John in just a minute. I have to also, of course, thank the patient participants, particularly posthumously our famous patient one, who we're trying to get at least 5 papers out of. We're up to number 3 right now, I think. So, so I'll thank you and turn the microphone back over to John, and he'll tell you what we can do about all of this. I can take some questions at this point. We had one in the back there.
Hi, Jason Hataye from Vaccine Research Center. It's just for Mary Kearney's experiment that you just showed, where 4 out of 384 were HIV RNA positive. So were those stimulated, or were they just like kind of resting?
No, they were completely. They were taken as with as little manipulation as possible directly from the patient. So they're taken from the patient, separated and frozen as quickly as possible, with deliberately no manipulation, not even warming them up to the possible point. So because we wanted to really be as close as possible to the actual state of the virus in the patient. So then that kind of manipulation experiments to do that are on the drawing board, but we haven't gotten to them yet.
Why did you choose chromosome 16?
Well, because of the MKL2 gene, so it homed in on this. So the important point, you'll see if you read the literature on this, you'll sometimes see people talking of finding sort of clonal integration sites like that in patients and talking about integration hotspots. That's completely wrong. That's the point I wanted to make. That's not an integration hotspot. It's actually a cool spot, but it's selection that determines it, makes it a hotspot in patients, because it's doing something that allows the cell to become much more prominent in the population than it was to begin with. And that's the real point that I wanted to make by doing it that way.
Okay. Well, we'll have time at the.
How long do you want to go?
Well, we're going home in 20 minutes, but you can go as
Okay.
long as you want to.
Okay. Yeah, till 6. Okay. Thank you, John. That was great. All right. So let's just talk about how hard it's going to be to cure. So why can we cure HCV? Block all replication of HCV, very potent inhibitors. RNA template, RNA genome lasts not very long.
No, get John's slides back.
Probably the half-life is such, I don't know the exact half-life, that all genomes are gone within 6 to 8 weeks. So,
Where's your stick? You have it in memory?
No.
That's what made cure HCV. All you had to do is block replication. Template disappears. How do we cure cancer? It's all in the CAR T cells. What's the key about CAR T cells? They recognize the stable epitope that continuously expressed in the malignant cell. CD19 finds what it needs, right? So for HIV, even harder than cancer, much harder than HCV, because the template doesn't disappear. There's always a hard copy of an intact provirus in some cell that has an indefinite lifespan, probably the life of the individual. Okay? Second, this is the killer. suddenly go silent, downregulate CD19. CAR T cells would be working. They would fade. Let's say could re-express 19 and go on its merry way, or proliferate in the absence of expression of 19. It would be invisible. And this is actually the mechanism whereby some B-cell lymphomas escape CAR T cells. They either change the CD19 epitope by some splicing variant, or they downregulate expression. Look at HIV. John told you that most of the reservoir is in clones, and that most of the proviruses are silent. They're not expressing. So there's nothing we know of, no means we know of how to get them to express, to expose themselves to the immune system so that we can kill the infected cell. So it's a monumental problem. You have a permanent copy. You have the ability of the virus to be silent, invisible to the immune system. So we have to conquer both of those problems. And B-cell lymphoma looks relatively easy compared to HIV. Okay, short history of cure research. It was thought with the advent of antiretroviral therapy and imaginative mathematicians, and based on the early decay of plasma RNA, that you would get to zero and eradication. Okay? That lasted all of about a year. When 3 labs, the Fauci lab, Clonal lab, Richmond lab, all discovered cells that were infected with a provirus that could produce infectious virus, but they were latent. Okay, so the latent reservoir was discovered, and that set the tone for 12 years or so. That cure was impossible. Okay. By the way, I believe it was Aristotle who said that nature abhors a vacuum, and influenced science for 1,000 years, and led everybody to believe that space had nothing in it. And from what we know today, nothing could be further from the truth. Space is loaded with important atomic particles and energy. So this is a relatively short period of time. Okay, surprise, Timothy Ray Brown had acute myelogenous leukemia and HIV. He got 2 bone marrow transplants for his acute myelogenous leukemia, and many years later has no detectable HIV. What happened? How's this possible? Okay, and can we reproduce it? So here's a cartoon I'm going to show you over and over again. You have latently infected cells and chronically producing cells. The chronically producing cells are in a minority. The latently ones that are a majority. They produce virions. Timothy Ray Brown got total body irradiation, which is lethal to lymphocytes. He got antithymocyte globulin, another killer of lymphocytes. He got graft versus host disease, and these killed and replaced all the infected cells and his entire hematopoietic system with donor. He got transplanted by Hero Uther, very clever clinician, who said, let's transplant him with a donor who lacks the co-receptor for HIV. And it just so happens, this was a lucky situation, Tim Brown did not have virus in his body that was X4 tropic. Could not use the alternative receptor, CXCR4. So cells could not be infected by his virus. He had no X4 virus, and he was cured. Now, the sobering news is the mortality from the bone marrow transplant he underwent, which was allogeneic, is about 25%. But that was overlooked. Okay, it was an important event, right? I think I reviewed the case report 2006 or 5, and none of the reviewers believed it. They said, sent it back, said, wait a couple of years, wait a couple of years. And so finally in 2009, it was published. So this is primarily inspirational, and it's galvanized a worldwide effort to cure HIV infection. The problem is, hasn't been reproduced. The number of people cured is 1, and it's been 1 since 2009. There were some near misses, but reality has set in. So 2 Boston patients got the same procedure Timothy Ray Brown did. They got transplanted with a CCR5 wild type homozygous donors. At the end of the transplant, they had undetectable HIV DNA in the blood, and less than 0.001, less than 1 in 100,000 of cells by the most sensitive assay was their original host cell. Okay, so normally you have 1 in 1,000 cells infected. Now we've reduced that by another 100,000. So the reduction in the reservoir is somewhere between 10 to the 8 and 10 to the 9. Okay, that emboldened, and I would have done the same thing through informed consent, to stop antiretroviral therapy in these individuals who had been suppressed for 3 to 4.5 years. The first one relapsed in 84 days. The second one in 210 days, 224 days. Okay? So that means that we're still, despite complete, what the hematologist would say is complete chimerism, meaning complete replacement of the hematopoietic system by the most sensitive assay, there was still a host cell with an infected provirus that survived that. Okay? No other individual transplanted, even with a homozygous Delta 32 positive donor, Delta 32, positive homozygous donor has survived. Unfortunately, they've all died, and one of them died because CXCR4 tropic virus emerged and led to CD4 depletion. So the idea that we can eradicate from the human body all HIV-infected cells that could produce infectious virus is a pipe dream as far as I'm concerned. It's not going to happen. Okay? So what do we do about that? Well, there was enthusiasm that if we treat people early enough, we get them right, at the moment of infection or slightly thereafter, the reservoir will be small and will decay down to nothing. Sorry, no good. Okay? Here's Chintanat Warren's data. 7 men, 1 woman, treated with ART in FEBIG-1. This is just RNA positive, no antibody, no P24, okay, because the level of viremia is below the limit of P24 sensitivity. No immune response, treated. They all relapsed. 1 held on for 8 or 9 weeks. The rest rebounded very early. Okay, so early treatment isn't going to do it. So what is going to do it? So from the Berlin patient, from Timothy Ray Brown said, some scientists said, we'll knock out CCR5, okay? And that'll cure. If we convert somebody's immune system to CCR5 homozygous negative, virus can't replicate, okay? Alternatively, reverse latency, so-called kick the provirus out of latency, and then kill the infected cell that's now exposed to the immune system. Which one? Well, host cell modification. 2 companies, Calimmune and Sangamo, are trying that. Sangamo's knocking out CCR5. Calimmune's knocking in 2 antiviral cassettes, short hairpin 5-RNA and a fusion inhibitor, C46. Okay. The Sangamo group has modified CD4 cells, zinc figure knockout of CCR5, reinfused those. They constitute about 14% of circulating CD4s. They last about 48 weeks. With ART interruption, they are selected. They increase in frequency. And in one of the 4 patients who they did this on, shown on the next slide, patient 205, virus didn't rebound. Now, it turns out this individual was heterozygous for Delta 32 to begin with. So it was halfway there, and then with knocking out cells ex vivo and reinfusing them, the target cell number was such that viremia did not return after interruption. It was controlled. So I think this is really interesting, and there are other instances, at least in a monkey model, where you may not need to protect every CD4 cell from HIV infection by knocking out CCR5. So it's something that's interesting and should be pursued. Challenges moving forward is they had to make room for the CCR5 knockout cells, you have to give cyclophosphamide or busulfan. And let me just say this, that the clinicians who see patients in clinic say, John, you want to do what? These people are living a normal life. Wynn said they could live to be 50. They can live to be 90 on antiretroviral therapy, 1 pill once a day, and they see the doctor once a year. Okay? I see my cardiologist every 6 months for a variety of ailments, okay? And I take more than 1 pill once a day. And most people my age do, okay? Right? So should we be giving them cytotoxic therapy? Will there be CXCR4 escape? I think that's a major problem with this strategy, because unlike Timothy Ray Brown, most people who are started on ART late, CD4 less than 200, will have a dual tropic or CXCR4 tropic virus. And is it scalable, and what's the cost of doing this type of therapy? Those are key issues. Okay, so let's look at the kick and kill. Here's the cartoon. You induce latency reversal. You then stimulate immune effectors, CD8 positive CTLs, NKs, macrophages, and possibly in combination with antibodies to target OMV-expressing cells to kill that infected cell that's producing virions and eliminate it before it infects another cell. That is a pretty high order. John, how long do you think envelope is expressed on the surface of a cell before it starts making virions? Maybe 4 hours? Maybe 1 or 2. So the window of opportunity to target that cell is very narrow. So you have to have these troops ready to go, circulating constantly, and kill any cell that starts to express envelope before it can produce enough virion to infect another cell. That's a high bar, okay? All right, I can't go through all the kick and kill candidates. They're LRAs. I'm going to show you some initial data from an HDAC inhibitor called romidepsin, a TLR7 agonist, okay? A engineered antibody called VRC01, okay? So a kick agent, a kill agent, and people are talking about immune checkpoint blocking antibodies, the resolutionized cancer immunotherapy. Cellular therapies are being talked about, including CAR T cells. And then I'll end up by talking about viral vectors that express mosaic antigens in a monkey model. Okay, so romidepsin is an HDAC inhibitor that in the laboratory causes this latency reversal. We did a trial in the AIDS clinical trial group. We chose romidepsin because on a per weight basis, it is the most potent HDAC out there. We carefully chose the dose. The maximum tolerated dose is way above this it's a drug used to treat lymphoma. It's 14 mg per meter squared, given in 3 cycles. we either gave 0.52 or 5 mg per meter squared. And we did this because ex vivo cells from patients, we could easily induce HIV expression. Okay. And so we studied these 3 dose levels in a blinded trial. Okay. And here are the results. This is the level of viremia. Nothing happened. Here's the level of cell-associated RNA. Nothing happened. Here's the romidepsin concentrations. They're dose proportional. So we got exposure, but no pharmacodynamic effect. We're not going to give up, and we're giving multiple doses at the highest dose, 5 mg per meter squared, in a 4th cohort that's fully enrolled. Okay. How about antibody? One of the first antibodies that came out of the VRC was VRC01, broadly neutralizing, not as broadly neutralizing as the later generations. I called up John Mascola and said, why don't we give this high-dose antibody to HIV-infected individuals on chronic suppressive ART and see whether it perturbs the reservoir? We did this fancy randomized trial with staggered dosing here and here in different arms, which allows you to compare pre and post across 40 patients. We looked at just about every virologic endpoint we could think of, including cell-associated HIV RNA to DNA, cell-associated RNA, cell-associated DNA, stimulated virus production, and plasma viremia. Everybody said, oh, why bother looking at plasma viremia? This antibody is just going to glom onto that virus and clear it like that. Well, guess what? No change in any of the endpoints, and what was surprising to everybody, including me, was no change in the level of viremia. Did not affect viremia, this high dose of monoclonal antibody. So what's going on? It was safe, well tolerated, didn't affect any virologic parameter. So you have to explain why. Well, my favorite hypothesis is that the virus that persists in an infected individual and has escaped all the endogenous antibody may be resistant to VRC01. Okay. Second, there's something about VRC01 or NK cells or the reticular endothelial cells that fails to clear OMV-expressing cells, or VRC01 doesn't get to the right sites of virus expression. Other antibodies will be tried, but this was clearly disappointing. So we couldn't reverse latency. We couldn't impact the reservoir with a designer antibody. In untreated infection, it knocks down viremia by a log to log and a half, and that's probably because it's blocking viral spread. It's actually neutralizing virus that is free and it's acting as an antiviral. Whereas here, we thought it would act on the chronically infected cells that are expressing envelope and promote their killing. It didn't. But also was surprising, it didn't clear the little bit of residual viremia that we can measure. Okay. So what about stimulating the innate immune system with a TLR7 agonist like GS9620 that Gilead's developing? It could reverse latency, and it could stimulate innate and then subsequently adaptive immune responses. Here's just the internal signaling pathway of the TLR7 shown here. This study looked at multiple dosing done by James Whitney at Beth Israel at different dose levels. Suffice it to say that blips in viremia were produced, and when treatment was interrupted, 2 of the monkeys that had gotten either 0.1 or 0.15 mg per kilogram of either a preclinical compound or a clinical compound were cured of their SIV infection, and virus could not be recovered after sacking these animals and injecting new monkeys with tissues. No virus grew out. Yes? Orally. Yeah. Let's go back. So 10 doses and then 9. So multiple doses. Okay, and so there's a human trial at our site and a few others looking at a dose escalation study, 6 doses, randomized trial. We're finished the 4th enrollment, 4th cohort of 6 milligram every 2 weeks for 6 doses. And I won't tell you the result, but cohort 5, 6, and 7 are planned. So that tells you something. Okay. And I'm going to end on a positive note here. This is the macaque study. Looked at a combination of TLR7 agonist, this tool compound, and a Gag-Pal-Omv vaccine at 26 prime MVA boost. This was published in Nature from Dan Baruch's group, 4 groups of animals, add 26 MVA alone, with add 26 MVA with the TLR alone, or sham. And I'll cut to the chase. 3 of the 9 animals that got both failed to controlled and failed to rebound. And studies are ongoing to see whether there's any persistence of virus. Okay, so how does the future look for HIV cure? The bar is very high, especially for eradication, and one that would be effective in all HIV positive individuals. There are still fundamental knowledge gaps, specifically brain reservoirs, which in my mind is the final frontier, and what immunologic correlates there will be of viral control off ART. I think a few more cures will follow, defined as control of viremia off ART in specific populations first. And if I had to guess, it would be early treated, given a TLR agonist, with either a vaccine or a broadly neutralizing antibody or both. And there are always unrealistic optimists and incorrigible skeptics. But just remember, 1 pill once a day to treat HIV infection was once a fantasy. And so if you had said in 1987 that we're going to be able to control HIV with one pill once a day, they would have done a psychiatric evaluation on you. So lots of people to thank, prevention, teams, large teams in the ACTG for both the studies I showed you, and a great group of collaborators, Pitt, ACTG, John's Drug Resistance Program, Leidos, Tufts, Gilead, the Gates Foundation, and NIAID.
Fantastic. Thank you both for an incredible afternoon of really state of the art. There are a few questions before the fire marshal tells us we have to leave.
Jason.
I kind of had this question I've been meaning to ask Dr. Coffin for a while. So there's these restriction factors like the ApoBec that you referred to and showing some of your sequencing trees. And so it can hypermutate some of the genomes, but it doesn't do it in all of them. And what's known about, like, why that why do they only get a small fraction of the viruses?
We don't know that for sure. There are a couple of hypotheses. One is that ApoBec 3G and 3F in particular are very strongly counteracted by a viral gene called VIF. It may well be, because the virus does accumulate mutations, that during the accumulation of mutations, you occasionally get a virus that's defective in that particular gene. And therefore, when that virus gets into another cell, or actually has to go through two such cells for reasons that are a little complicated to discuss. But nonetheless, so that it may be, in fact, the VIF gene in these viruses is defective because of a pre-existing mutation and therefore can no longer counteract APOBEC. It may be that some cells express such high levels of APOBEC that they cannot effectively counteract. The virus cannot effectively counteract it. So it's something like that is going on. It's an antiviral. It's a viral defense, antiviral defense system that sometimes fails on one side or the other. I should note that there's a very strong selection for that for mutations like that because those cells survive the infection because the provirus is very highly defective. The enzyme changes at a very high frequency, changes C's to U's in the minus strand, leading to G to A mutation. APOBEC3G, in fact, for example, a particularly good target site is a tryptophan codon, of which there's only one, and it turns every one of those into stop codons. So it's a very effective, lethal antiviral device that probably actually, just to go on a little bit, is probably what protects us from retrovirus infections from other species. It's probably why XMRV is not a human pathogen, despite what was passed around a few years ago, is because we all express this protein, this antiviral protein, as part of our defense mechanism.
So what do we know about the brain? Are all cells in the brain susceptible to HIV?
No, not all cells. It seems like a cell called a microglia that has a myeloid origin is infected. We have some really interesting data that will be presented at CROI that we don't detect free virus in the spinal fluid, but in about half of people after 8 years of therapy, we could still detect infected cells in the spinal fluid. Proviruses, and the detection of those infected cells in the spinal fluid is associated with neurocognitive impairment. Okay? And that was a shocker to us because we're talking about a handful of cells in the spinal fluid, and so that suggests there is a brain reservoir and it's clinically relevant. And my great fear is just that we can maybe attack the reservoir in lymphoid tissue, but every infectious disease that I know of is more difficult to treat in the brain than it is in the periphery. I mean, that's the 8 years on suppressive ART. I mean, and the elite suppression, no blips in those 8 years. So that, in my mind, the brain is going to be one of the final frontiers of the cure effort.
Does the simian virus affect the brain?
Not as much. You have to get a neurotropic variant and co-infect with a lymphotropic variant. Janice Clements has a model of neuro AIDS in the monkey. It's a complicated model.
Well, listen,
I guess continuing along with the microglial infections, there are different profiles of microglial markers and inflammation. Do you know has there been any studies done on saying looking at the profiles of infected microglial cells and especially in terms of hand diagnoses?
So the really difficult and I think insolvable problem with looking at the brain is you can't go around taking pieces of it. You can't take pieces of it.
You mean in humans?
In humans. All right?
Well, that's not quite true because, there are groups here who are making organoids from brain biopsies.
Maybe gotten post-mortem the brain, but in healthy people, getting pieces of brain tissue for science is going to be incredibly difficult, if even deemed ethical in certain situations, which I doubt it would be. I mean, how many in the room would volunteer for a piece of their cortex or basal ganglion to go into the freezer? I certainly wouldn't.
That's for science, guys.
Come on.
Well,
So that's the biggest obstacle, Michael.
on that sobering note, thank you very, very much. This has really been a